Hi, everyone. Welcome to OsteoBites. Thanks for joining us today. We'll give a couple seconds for everyone to get settled in our virtual room. We've been on an extended break, I think since late May, so it's really good to be back. My name is Christina Iptoma, and I am mom to Osteo Angel Dillon and Director of Scientific Programs at MIB Agents. And today on OsteoBites, we are joined by Dr. Olivier Elemento, from Weill Cornell Medicine, and he will discuss his lab's precision medicine research, which combines big data analytics such as high performance computing, mathematical modeling, artificial intelligence, and machine learning with experimentation to develop entirely new ways to help prevent, diagnose, understand, treat, and ultimately cure disease. Thanks so much, Dr. Elemento, for joining us on OsteoBites today. We are thrilled to have you. And thank you also to our panelist, Daniel, for joining us. Everyone, please feel free to submit questions during the presentation through the Q&A on Zoom, and we will share those with Dr. Elemento. A little bit more about our guest today. Dr. Olivier Elemento is a professor of physiology and biophysics at Weill Cornell Medicine, and since 2017, he has been the director of the Carol and Israel Englander Institute for Precision Medicine, a large multidisciplinary institute that uses precision medicine technologies and informatics to uncover the molecular mechanisms of disease and individualized disease treatment and prevention. He is also the associate director of the Institute for Computational Medicine, director of the Laboratory of Cancer Systems Biology, and co-leader of the Genetics, Epigenetics, and Systems Biology program in the Meyer Cancer Center at Weill Cornell Medicine. His research group combines big data and artificial intelligence with experimentation and genomic profiling to accelerate the discovery of cancer cures. I'm super excited to have you here today. And just a couple of quick announcements before we get started. We, I think, are still riding the high from our Factor conference in Cleveland a few weeks ago. It was really great to bring together our parts of our community, researchers, clinicians, patients, families, advocates, industry, and funders to one place for a full-on educational and collaboration fest. So we'll be sharing abstracts and photos soon, so stay tuned. And we will also be announcing our Factor 2025 location and dates shortly. We also have our next virtual tumor review board for osteosarcoma, which we call Turbo. It's scheduled for next week, July 17th at 5 p.m. Eastern. Turbo is open to clinicians and researchers with an interest in osteosarcoma. And we have a full agenda already for our meeting next week. But if you'd like to be added to the calendar invite, if you're not on that already, or if you're interested in submitting a case in the future, I'll put a link in the chat to a form that you can submit. Daniel, could you please introduce yourself?
Yeah, hi everyone. I'm Daniel. I'm a part of the Junior Advisory Board this year. I guess first of all, I just want to echo what Christina said about Factor. I went this year and it was an amazing experience, just for the scientific work being presented and just being with the entire community. I'm excited to hear what Dr. Elemento has to say today about AI and personalized medicine. I have to admit, I've had a lot of success with it for my computer science classes, so I'm particularly interested to hear about how it can be used for osteosarcoma. I think that's all I have to say, so I'll hand it off to Dr. Elemento.
All right, thank you so much. Thank you, Christina. Thank you, Daniel. I'm delighted to be here today. I'm going to share my screen so that you can see my slides, and I'm going to start a presentation. And I'm going to start by saying that we work on trying to invent the future of medicine and try to make it more personalized. Our work is not just focused on one cancer. We focus on the process by which we can envision a form of medicine that's much more tailored to our disease, to the disease that patients have, as opposed to kind of trying to treat the average patients, which is what medicine is still largely doing right now. So what we do in our institute at Weill Cornell is really to combine different types of technologies to rethink how we analyze clinical samples, in this particular case, cancer samples, and really try to understand the biology of every patient, understand what's driving the cancer within every patient, and come up with ideas about how to treat patients. So I'm not gonna talk much about osteosarcoma or even sarcoma in this talk. I'm gonna try to illustrate what we do with other types of cancers. But everything that I'm saying is completely applicable to sarcomas and also osteosarcomas. So here's my talk. I want to divide it into multiple different sections. I'm going to start with a brief overview of precision medicine. I will talk about DNA sequencing, which is still the main way by which we can understand cancer, because it's focused on trying to find the mutations, and the mutations are specific to every patient within cancer cells. But I also want to highlight something that's critical and applicable to osteosarcomas, to sarcomas, and to every other cancers, which is that, in fact, when you look at cancer under a microscope or using the technologies I'm going to show you, you realize that cancer cells in a cancer are in fact a minority of cells. In fact, in many cases, you have all kinds of different non-cancer cells there. In the past, I think the researchers, scientists, clinicians have kind of discounted a little bit, these non-tumor components of tumors. I think what we are seeing as we analyze lots and lots of cancer samples is that this component is really important. And I think it has a great future to be used as a way to stratify patients, but also as a way to find novel targets that could be used to better treat cancer. The last part of the talk is focused on a different angle for our research, which is really focused on this idea that we can better understand cancer by maybe perturbing cancer cells and asking more questions about the cancer this way. The techniques that we use to assess cancers in general are pretty static. You kind of get a snapshot, but obviously we know that cancers are complex, and we'd like to be able to kind of poke the cells of every patient to kind of see what else they tell us about themselves when you do that. So that's for the last part of the talk is going to be focused on that. So as I said, our institute is focused on this idea that we think that we can make medicine more personalized than what it is. And why does it matter in the context of, for example, sarcomas? Well, we can take two patients with the same disease according to how we describe disease. Here in this case, it's two patients with leiomyosarcoma. The pictures are not real patients; they're just stock pictures. But the data is real. It's from real sarcoma patients. So these are osteosarcomas, but a different type of sarcoma. You are seeing here two lists of mutations that were obtained after sequencing the DNA of these two patients. They have distinct, if you see the number of mutations which is shown here, there's actually pretty distinct features, right? This patient here has a lot of mutations in the tumor genome. This patient has very few mutations. One thing that's really striking when you look at the list of genes and this one patient and compare it to the list of genes and this other patient, I'm only showing the top of the list, but it just gives you a sense. There's no overlap in terms of mutations. So what's causing the cancer, we know that now the mutations are causing cancer. What's causing the cancer in those two patients, which have the same disease according to how we describe disease, is in fact very different, right? Mechanisms are different. And therefore, we think that they should be in fact labeled differently. There should be different disease, right? If you were to rethink the labeling of cancer, you would probably not name those two patients or those two cancers with the same name. You would name them or describe them based on the mutations that they have, and the mutations are different, and therefore there should be different disease. That's really what underpins this idea of precision medicine, which is that we need to understand every patient so that we understand the mechanisms that cause disease, and then go after the mechanisms themselves. Try to use those mechanisms to kill the cancer cells in a personalized way. And so that underpins everything that we do in our institute. Really this idea that every patient develops a different disease and a different cancer. Medicine has always been personalized, right? There's no, I think it would be misleading to say that it hasn't. But on the other hand, I think now what we can contemplate, and that's only possible in the past few years because of technological innovation, is a much, much deeper ability to quantify what's happening within cancer cells using technologies such as high throughput DNA sequencing, which is what you see here, allows us to sequence cancer genomes in a very short amount of time. This technique here, this machine here, allows you to take a snapshot of tissue at single cell resolution. We'll go back to this. You can basically see the cells within the tissue, within the tumor, almost understand the battlefield of cancer, how the good cells and the bad cells are battling each other in real time, if you want. It's not dynamic, but it's a snapshot. But it allows you to see things that normally you don't even look at. And then you have this idea of avatars, which I also very much like. We focus on cell avatars, which is essentially creating ex vivo models of people's cancers. There's a lot of interest in going beyond cell avatars and thinking about in silico avatars, computational avatars, simulations of people's cancers, which would allow you to test many hypotheses, many drugs, combinations, and then treat the patients based on what you learn from the testing. So all of these directions, all of these components is part of our sort of mission, part of our vision about how we can bring personalized medicine to cancer patients. And I had one thing on the right side here, which is that all of this is enabled because these techniques generate a lot of data. You need to have something like AI to be able to integrate, analyze, combine all this data into predictive models that connect patient data with outcomes, predict response to therapy, predict combination therapy, predict diagnosis, better, predict relapse, and so on. So the analytical component is really critical as well. So everything we do is done in the context of a hospital. So we're not disconnected from the hospital. We get patient samples from the hospital here at Cornell. And our goal is really to analyze these samples and try to combine all the data that we learn from every sample into a report that is eventually under IRB or, in certain forms of sort of return, is returned back to the patients and is used as part of a clinical care. So we are very integrated into clinical care. We're not doing that. We're not doing everything that I'm going to show you divorced from clinical care. We're very much integrated into everything. So let me tell you a little bit about what is being done now here, but also in other centers. The analysis of clinical samples, especially cancer samples, is still quite very focused on DNA sequencing, the sequencing of the DNA within cancer cells, and the discovery of mutations. The sequencing of DNA in cancer cells has evolved over time. This slide here shows a little bit of sort of gives you a sense of the evolution of the field. What it used to be, and it's still done in many centers, that you would essentially look at a cancer genome, which is very large. The human genome is 3 billion nucleotides, and the cancer genome is about the same size. So many centers are looking at the cancer genome through a very narrow lens, looking at just a small number of genes that in the past have been shown to be connected to cancer. And we think that there's a lot of limitation to this approach. So that's why we've invested here at Cornell into two different technologies. One of them was whole exome sequencing, which is the sequencing of every single gene in the cancer genome, which is something we've done for many years. But since then, we realized that we need to essentially sequence the entire DNA of a cancer cell from beginning to end. I would tell you that that's really very much applicable to sarcoma and also osteosarcoma, because rare cancers don't really sort of benefit from the approaches that you see on the left and in the middle. And part of the reason why that's the case, rare cancers are not driven by common mutations, right? The mutations that you see commonly found in many cancers, and the ones that go into the panels, for example, on the left side, the one that go into the decision about which gene to sequence. Rare cancers are not represented often in those panels. The mutations from these cancers are not represented because they're not common mutations. And so essentially you end up with many patients that are essentially mutation negative according to targeted panels, and maybe even sometimes whole exome sequencing, because of the fact that the mutations that drive those cancers are not part of the windows that people look to assess mutations. That's why we do whole genome sequencing in a very broad sense here at Cornell, which allows us to assess every single mutation in the cancer genome, not just the mutations within the genes, but also mutations involving translocations, structural variants, combining different fragments of DNA or from different chromosomes, sometimes creating new genes, or putting a piece of, let's say, an enhancer or promoter next to a gene to disrupt the expression of that gene. So we're focusing on that very much in our institute. This really idea of seeing, this broad view of the genome.
Dr.
Just a
Elemento,
few.
sorry,
Yeah,
back to
of
that
course.
last
Yeah,
slide.
please.
What is that?
Yeah.
What is the coverage number refer to?
So the coverage number essentially corresponds to more or less how many cells you sample within the cancer. The idea of the coverage is, that when you sequence DNA from a tumor, you isolate the DNA from the tumor, and then you essentially sample the DNA. You can't sequence every single cell, right? I mean, that's still very difficult. So what you do is you combine the DNA from many, many cells together. You select a subset of that DNA, and then you sequence it. So the one way to think about the coverage is essentially how many cells per cancer do you sequence on average. And so that allows you to identify mutations that are not present in sort of just one cell, but mutations that are present in many cells in the cancer, which are typically the mutations that you want to find, right? If a mutation is only present in a very small fraction of the cells in the cancer, it's unlikely to drive a cancer, right? Because, the cancer is the totality of the cells. So the idea is that, we sequence multiple cells. So one upside of targeted panels is that you can essentially sequence more cells per cancer, but one downside is that you look at a very narrow window in the genome.
Got it. Thank you.
And so there's actually a lot of data now showing that this broad sequencing of a cancer genome is really beneficial. It finds many more mutations than this narrow focus on the genome. This is a paper that we published a few years ago now, showing in lung cancers, in the subset of lung cancers that did not have classical driver mutation according to targeted panels or whole exome sequencing. Whole genome sequencing was able to find very complex mutations, such as the ones that you see here, which we call structural variants. Essentially, these are lots and lots of bits of DNA from one gene or different genes that are kind of broken apart and then recombined into a new piece of chromosome that creates new genes or a new composition of the same genes, affecting sometimes important genes, but very difficult to find using traditional sequencing methods. So, and the analysis of this kind of data here requires not just DNA sequencing, but also complex analytics to be able to kind of piece back the puzzle together, because we are sequencing genomes, small fragments at a time, and so we have to reconstruct the genomes, like if you were to put a puzzle back together with multiple pieces. So it's a complex slide, but here it really shows multiple classes of mutations that were not visible using traditional sequencing techniques, but were visible using whole genome sequencing. There's additional classes of mutations that come from the fact of found by whole genome sequencing that come from the fact that when you focus on a narrow window in the genome, what you have to do is to pull down the DNA that is found in those genes. And that process of capturing DNA can be inefficient for certain genes. For example, genes that have a lot of G's and C's, the nucleotide G and C's, this DNA doesn't capture very easily because of the structure of the DNA. And so you lose mutations as a result of bad capture efficiency. I sort of showing this slide here, which is an analysis of our large cohorts of patients here across multiple different cancers. One of the nice things that whole genome sequencing gives you is not just mutations, but also patterns that occur as a result of finding many mutations across the entire genome, including outside the genes, between the genes. There are certain types of mutational processes. One of them is a mutational process called homologous recombination deficiency, which I like to describe as BRCAness, because it's basically the result of having a BRCA mutation. If somebody has a BRCA mutation and develop cancer, if you look at what the cancer looks like, the DNA of the cancer looks like, it has a lot of mutations across the entire genome, but these mutations have a particular pattern. They occur in a specific mutational context, in a specific sequence context, or they consist of copy number alterations or certain types of copy number alterations, for example. What matters here is not the mutation themselves, is the signature of the mutations across the whole genome. Why is that important? Well, we know now that many patients with, for example, all kinds of different cancers can have BRCAness in their genome, basically this mutational signature, even if they don't have a traditional BRCA mutation. And the reason for this is that there are probably many mutations in certain genes that are not BRCA, but kind of like BRCA or have a function similar to BRCA that create the same mechanisms of repair deficiency in the tumor cells and basically create BRCA-ness. So what the field has done, and we've done here also at our corner, is to develop an algorithm to be able to detect BRCA-ness in cancer genomes. What's really interesting is when we screen all kinds of different cancer genomes for BRCA-ness, we actually find many cancers that are not known to necessarily, have a lot of BRCA mutations, but actually have BRCA-ness in their cancer genome. So we have the same phenomenon as BRCA, as essentially having a BRCA mutation. It's critical because in pancreatic cancer and prostate cancer, for example, if you find such BRCA-ness, you can treat patients with platinum or PARP inhibitors, which are two classes of drugs that are known to be effective in the context of this mutational signature. So we actually found lots and lots of patients who, potentially could benefit from these therapies. Outside of breast cancer and gynecological cancers. And this is an example here of a patient where we found such phenomenon in the cancer genome. And was actually, as a result, treated with, in this particular case, platinum, and had a complete response to that therapy as a result of the treatment. Not just that, but because we sequenced the DNA of the cancer from beginning to end, we actually could find important mutations that we think are responsible for the BRCA-ness phenotype of that patient as well. So it's just a good example of how, we using whole genome sequencing, you can find unexpected mutations or mutational signatures that then can inform treatment and are very personalized to that one patient. So I'm gonna move on to the next topic of my presentation, which is what I was telling you earlier, which is that it's still kind of common to think of cancer essentially as just a collection of lots and lots of cancer cells, but now we know that most cancers actually consist of other things outside of cancer cells. Things like immune cells that go into the tumor, infiltrate the tumor, and try to fight the tumor, but are blocked from doing that effectively by potentially tumor cells secreting things to kind of tell the immune cells to shut down. Blood vessels, also very common in tumors. Fibroblasts, which kind of provide structure to normal tissue, also found very often in tumors. So the point here is that a tumor is a complex ecosystem of tumor cells and non-tumor cells. This has really in the past been kind of ignored by a lot of scientists to really focus very much on the tumor cells. And DNA sequencing is a good example, right? DNA sequencing is only finding mutations or looking for mutations and finding mutations in the tumor cells, but not capable by itself to tell us anything about the non-tumor cells of the tumors. So to study this complex ecosystem, we had to resort to other technologies. Technology that we use here are called spatial techniques. Basically allow us to take pieces of tissue, or let's say slices of tissue, thin slices of tissue, cut away from a tumor, and then analyze them in their spatial context. Essentially try to take pictures of the tumors at single cell resolution, and essentially kind of inform us on what kind of cells you have in the tumor, and also what's the architecture of tissue in the cells. I'm just going to show you a few slides showing some results for another cancer that we studied using this technique, and we're studying this cancer, but also many other cancers using the same technique. This one is a type of tumor that's a cancer of B cells, cancer of immune cells called B cells. So we have a large cohort of these cancers, so we're able to analyze tissue from many of these patients. You see here, this is a slide coming from essentially a cohort of multiple patients. Each dot is one patient, and so we can analyze many patients at the same time. This technique here is based on antibodies, and so we have to select which antibodies to look at. These are basically antibodies that recognize proteins. And so that allows us to essentially kind of light up where in the tissue you have expression of these proteins. And because some proteins are quite specific to certain cell types, for example, some proteins like CD4 are almost only seen in CD4 T cells, which are immune cells that are, have a specific function. If you see CD4 expression in a piece of tissue, you know that you have a CD4 T cell. There's, some markers that are quite specific to immune cells that are important. But we also have markers that are, for example, telling us which cells proliferate, which cells express checkpoints, which cells undergo apoptosis, which cells, for example, express HLA, which is the machinery in the cells that allows the tumor cells to be recognized by the immune system. And we have additional markers that allows us to assess the structural component of the tumors, like the fibroblast, and so on. So the analysis of data like this is, in fact, quite different from sequencing data analysis, DNA sequencing data analysis. It's actually much more computer vision. That's why we started using AI, because we actually have to use AI as a way to kind of find the boundaries of the cells based on the markers that are expressed. The markers that tell us what the nucleus of the cells is, where the membrane of the cells, where marker for all these different things. The point here is that we end up being able to essentially find where the cells are in the tissue. And then based on the combination of markers that they express, we can essentially associate each of these cells with a specific identity. So this is one patient here. This is raw data. So you see it's kind of messy with different proteins expressed in different places. CD20, that's a marker that's very specific to B cells. Only B cells express CD20. So if you see green here, it basically means that you have a B cell. The reason why you have little dark dots in the middle here, these are the nuclei. These are the nuclei of cells. And so there's no marker in this slide here. There's no marker for the nuclei. So you don't see the nuclei. You only see the cytoplasm. You see what's around the nucleus. But the point is that, through this segmentation that I'm showing here, and then there's the identification of the cells and the cell type of each of the cells, then we can go towards this picture on the right side here, where suddenly every cell has a very clear name. It has a very clear identity. So here, for example, if you see this blue cell here, this is actually a type of macrophage. This is another type of immune cells that has a particular function in tumors, is kind of trying to eat up the tumor cells when they can. Typically, it's disabled, but it's still there in the tumor cells. So this is actually a very now a very detailed picture, right? Because you can basically see, as I say, kind of a battlefield of cancer, right? So the green cells are the B cells, and most of the cells are malignant. So these are the cancer cells, the bad cells. But you can sort of assume that every other cell that is not green is a normal cell, and in theory, a good cell, right? It turns out that many of these good cells normally would be trying to kill the tumor cells, especially the CD4 T cells, the red cells, for example, but also the macrophages, and clearly they're not able to do so. And so now, really, the complexity or the next step of this analysis is to be able to relate this kind of pattern here that we get for every patient to specific outcomes. For example, does a patient that has this composition of the tumor prior to diagnosis or prior to treatment, are they likely to respond to chemotherapy, right? And so there's, that's really the critical work, and critical piece of the work that we do is try to relate this picture here to outcomes, try to combine this, simplify this picture, count maybe how many cells we have, how many B cells we have, or how many lymphatic cells we have, and then look at patterns describing each individual tumor, and then relate that to outcomes. One thing that we been able to do when using this analysis here and looking at all kinds of different populations of cells is to cluster tumors based on what you see outside of the tumor cells. We ignore the tumor cells for a moment, and we basically ask whether there are different types of microenvironment. This everything that's not green here is called the microenvironment of the tumors. So it has a particular composition. It varies from patient to patient. I told you at the beginning that the tumor cells from patient to patient, they may have very likely to have different mutations. It turns out that if you look at the composition of the non-green component of the cells from patients to patients, there's also quite a bit of variability. The variability is actually clear, but it's also not infinite, in a sense that we actually find subgroups of patients that have a very similar composition in terms of the microenvironment, in terms of the non-tumor cells. I'm not going to go into details about these different types here, but you can see and convince yourself that they are quite similar, different from each other. Now, the question is really, does it matter, right? And the answer is really that it seems like it does, because when we try to relate the composition of the microenvironment to survival of these patients, essentially how they respond to chemotherapy, we see a lot of variability. Right? We see, for example, that this group of patients here, which we call cytotoxic for reasons of the composition of the tumors. Those patients, when they have this microenvironment prior to treatment, they actually, unfortunately, they don't do very well. They tend to pass away reasonably quickly. As opposed to other patients which have a different microenvironment, which do much better in terms of response to chemotherapy. These are what we call survival curves. What this means is that you essentially group patients into different bins, into different groups, and you look at how they behave over time. Every time the patient dies, the patient passes away, there's a little decrease in the curve. So if you have a quick decrease like this, it means that patients pass away really quickly, and then the ones who don't tend to survive longer. So this is overall survival. This is progression-free survival. Essentially means time to relapse. Here, the decrease is relapse, as patients the disease coming back. And as you can see, we have a similar pattern, similar trend in terms of survival here. And the cytotoxic group or microenvironment also does not do very well, and the tumors come back reasonably quickly compared to the other subtypes. What we've done is actually to go beyond this, to combine all kinds of different features about the microenvironment, about the tumor cells, about the patients themselves, and essentially make an AI model that can predict survival in this disease with surprisingly good accuracy. We can basically predict by looking at this picture that I showed you earlier, those sort of circle discs of cells, which patients are going to respond to chemotherapy with surprising accuracy. And again, most of the information here is coming from the microenvironment, from the non-tumor cells. And so it's just, one, it gives us opportunities to maybe stratify patients and kind of, think about patients who are not likely to do well upon chemotherapy. Maybe they can enroll into a clinical trial. Maybe we can find other ways to treat them. But also, we can also look at what the model is telling us, right, in terms of the cell types or the components that are most important in the microenvironment, and maybe design new drugs that could potentially target those mechanisms, maybe to try to make the microenvironment a bit more prone to chemotherapy working, or maybe to completely, just use the microenvironment to target the tumor cells, themselves. So again, this is I showed you an example for a different type of cancer, but this is really applicable to every cancer. We're very much pan-cancer in our view, and everything that we do is meant to be applied across cancers, and just some projects are moving faster than others, but that's sort of where we are. Let me just show you briefly another angle of our research, which is the same. The goal is really to try to understand every patient. One idea that we've had for some time now is to try to think about how we treat patients and understand the limitations. Right? When we treat patients with a targeted therapy or chemotherapy, we can treat them one drug at a time. In many cancers, unfortunately, we cannot treat patients with more than, let's say, one or two drugs. The patients become too sick, and it's just very hard to test many drugs before finding the one that works. So that's driven our research to kind of try to think about how it would be possible maybe to test many more ideas and hypotheses on patients without having to treat the patients. So one idea is that essentially maybe what we can do is to try to build an avatar of a patient's disease by taking cells from the patient, growing them outside of a patient's tumor cells from the patients, growing them outside of a patient's body, essentially make a lot of mini tumors that are copies of a patient's tumor. The way we do this is to try to preserve the biology of the tumors from the patients. You have to try to make the avatars as similar to the patients as possible. We do this by growing the tumor cells we get from patients in a three-dimensional fashion. The idea is to kind of create little domes of gels, like you see here, that kind of mimic a little bit conditions that you have in patients in terms of matrix, in terms of architecture. We seed the gels with tumor cells from individual patients. Then we let the mini tumors grow in 3 dimensions within those gel domes. And you end up with things like you see here, where every little gel dome here contains, somewhere along the lines of 100, maybe 200 different small tumors, and they're all kind of growing, by themselves. They form 3-dimensional objects, if you want, or let's say masses of cells. Before I show you some of those pictures, I want to show you this, which is, again, highlighting the fact that we try to do this for every cancer. And so, this is not sarcoma specific. That's not colon cancer specific. It's across the board that we try to grow these avatars for patients here. Basically what they look like. This is kind of zooming into some of these 3-dimensional little kind of masses of cells and showing you what they look like. They look quite different from tumor to tumor, as you can see here. They actually look also different from patient to patient, but I'm not showing those slides here. But each of these little kind of balls of cells here, essentially, we think of it as an avatar of a patient's disease. And the idea is that we can essentially collect many of them at the same time. We can grow 20,000 mini avatars for every patient. Yeah, so that kind of gives us, a lot of, let's say, sampling of a patient's tumor cells. And the idea is to try now to kind of see how we can maybe, apply perturbations to those cells coming from patients and see how they react. Obviously, one perturbation that makes a lot of sense in cancer is to try to find perturbations, basically drugs that can kill the cells. Can we shrink those tumors, in a dish with an indication that maybe, anything that shrinks the tumor in the dish could potentially help the patients as well. So we do this. We have to do this using robotics. This is not something that we can do manually. We have to use a robot to do the testing of hundreds of different drugs. And so we built a very customized robotic system to allow us to treat these tumors growing outside of a patient's body using many, many different drugs and combinations. I'm just showing you here 2 different patients. This is colon cancer. So this is, again, just examples, but results of drug screens. What you see here on the x-axis are compounds. These are different compounds, different drugs. On the y-axis here, you have the efficacy of the compounds in this particular patient. And the idea here is to look at negative z-scores. So the drugs that are most efficient in this one patient here, are the drugs that are here in the bottom. So one thing that you see here, for example, for this one patient with colon cancer, trametinib, which is a MEK inhibitor, is quite effective at killing the tumor cells of this one patient. On the other hand, if you look at the patient at the bottom, which who has the same kind of cancer, according to how we label cancer, you see the trametinib is not doing anything to the cancer cells of this one patient, right? On the other hand, you have neratinib, afatinib, different drugs that are effective. So just highlight also the fact that from patient to patient, response to these drugs ex vivo is also quite different. And I think it also is one thing that's humbling about this picture here is that we do sequence the DNA of these patients and have information about the tumor cells and the DNA of the tumor cells. And to be honest with you, there was not much information from the mutations in the patients or DNA cells of these patients that could tell us that these drugs that we found to be effective actually were going to be effective. So we still lack the ability to efficiently read genomes, cancer genomes, and say this drug is likely to work. We are not yet as good as we like when it comes to doing this analysis.
And Dr. Elemento,
one thing to highlight here. Yeah,
of course. Sorry, just question on the organoids. So do those include also all those other TME cells in addition to the tumor cells?
Excellent question. And so everything that I've shown you so far does not. But our institute is very focused on essentially kind of addressing directly your question, right? So I did spend quite a bit of time telling you that the tumors consist of many non-tumor cells, and it's important, obviously that when your question is very logical in that as a follow-up. So I'm not showing it here, but for as many patients as we can now, we also isolate from the tumors non-tumor cells like immune cells, like T cells, for example. We also expand them. We try to make as many as we can. And then we take the tumor cells and the immune cells and we put them back together into co-cultures, into essentially kind of combined cultures. And we actually have shown that this allows us to basically kind of identify immune therapies that could potentially be effective by targeting the T cell to then kill the tumor cells. So it's more complicated, not something that we can do in a very routine way, but this is very much the direction that this whole project is really taking, is try to make the avatars as similar to what you see in the patients as possible. This is just showing you combination screens here. One way by which you can think about the future of cancer therapy is really, to think about combinations, how to combine drugs to effectively kill cancer cells in every patient. So this kind of screening that we do allows you to also identify combination therapies. These are examples of combinations that work well here, like trametinib and celecoxib. Just 2 more slides to show that we are doing work in sarcoma as part of this project. Our director of our organoid platform, Ben Hopkins, has been involved in several studies in sarcomas. This is one, myxofibrosarcoma. In this study, they actually found, using exactly everything that I've shown you so far, this was from one patient here, but they found new potential candidate therapies that were effective at killing the tumor cells of one patient. They actually, in this particular case, also identified the therapies. One of them was a CDK4/6 inhibitor. And then we're able to show that combining some of these drugs was giving rise to even better efficacy in terms of killing, obviously, you can see here. So these techniques, even though I've shown you very limited data on sarcoma, are actually quite applicable to sarcoma. And we'd love, actually, in fact, to be able to do this and more often in sarcoma patients. We haven't focused on this patient population very much, but we'd love to be able to do this. Just one more slide, actually, just to show you that our goal is that eventually to be able to build AI models to predict drug sensitivity just based on molecular information that we'd be able to measure. One proof of concept is this here. This is actually a colon cancer project, but we've actually done a lot of drug screens in colon cancer for lots and lots of patients. And we actually also have transcriptomic information, gene expression profiles of the tumors. And so we're able to show that we can essentially, now that we've done a lot of screening, we can take a new patient, put that new patient into the context, into this universe of patients where we already have done a drug screen, and then find the patients that are closest to this one patient. And then say, well, if in the patients whose gene expression profile similar to our patients or our new patients, you see a lot of patients sensitive to a particular drug, maybe that new patient that is very similar, maybe should respond to that particular drug. And this is here showing you proof of concept for topotecan. That's a chemotherapy drug. But we're able to predict which patients is going to be sensitive to topotecan based on the existing drug screens that we've done across lots and lots of patients with pretty good accuracy. Just an example. This is using a type of machine learning model called nearest neighbor, k-nearest neighbor, some type of prediction. So I'm going to stop here. I was trying to give you an overview of the different techniques and technologies that we use to personalize medicine. As I said, everything that we do is kind of cancer agnostic in some ways, very much applicable to every cancer. We think it's the combination of these technologies is the future of cancer therapy. It really highlights how we need to understand better every patient so that we can treat them better. But this is just the beginning, and we're trying to make these technologies as accessible as possible eventually for hopefully every patient, every cancer patient. I will stop here. I'd be happy to answer any questions that you would have.
Thank you so much. Lots to think about. Everyone, if you have any questions, please feel free to put them in the Q&A, and we will make sure to ask Dr. Elemento. But maybe I'll just kick us off with a couple. So, at least for maybe the past few years, I think when there was this kind of, move to precision medicine and all the potential of precision medicine, it's still kind of more in thinking about subtypes. It's like, breast cancer is a good example, right? Where there's we've kind of determined there are these different subtypes and there's different treatments for those subtypes, and that's been relatively successful. And I feel like even now, like in some of the clinical trials, they're more like basket trials, right? Where it's like, if you have this mutation, you get this type of treatment. But it's kind of one of those situations where it's like, the more we know, the less we know. And the more data that we have access to, like whether it's, the tumor microenvironment, the metabolism, like all these different factors that could be kind of affecting all these different interactions. It seems like that it is potentially so unique to that specific individual, especially if you take into account things like pretreatment, if someone's like a relapse patient. And so I'm just wondering, like, when we talk about precision medicine and personalized medicine, like, will AI really help us get to that point where it is really unique to that specific individual and potentially at different points in time throughout their treatment cycle, because their sensitivity to certain drugs may change after they've had another drug before that. So like, will we get to this point where it's aside from just kind of being able to bucket people? And even, I mean, it's for sure helpful, like you had talked about, like the 5 different types of the TME
Right.
characteristics, but where it's like in some of these diseases where it's so heterogeneous that really it's got to be at a super individual level.
Yeah. I think that's a great question, right? So my view is really that, I mean, thinking in the level of every individual patient is really the future of, medicine. I mean, the part of the question is, how to establish this paradigm, how to demonstrate that's really the case and that we can improve outcomes in thinking in this way. And I think that's, obviously being challenging, because part of the reason why, we are putting patients into groups is that this is really kind of meant to enrich, patients into, for example, clinical trials, right? So that, we can have enough patients to do a clinical trial and get a, readout. But I mean, part of the complexity of this is that, it doesn't, I mean, it's a kind of a convenience sort of type of process, right? But it doesn't really reflect, what's happening in patients, right? And that's not just for cancer patients, right? I mean, there's, there's obviously this outside of cancer, there's all kinds of different diseases that are considered to be rare disease that, maybe are found, in a very small numbers. And so we kind of need to think about how we can, address these rare disease patients. And I think the thinking there, I think it relates very much, to how we should think about cancer, which in fact, I think of cancer as essentially as a rare disease. Every patient is a rare disease. And so the paradigm that we've been using to kind of try to put patients into bins, I think, has some fundamental limitations. It just kind of simplifies the biological description of the patients, kind of, maybe sort of makes us ignore, the biology that's unique to every patient. I mean, I think what we have to start thinking about is, how to connect biological features with response to therapy. And that connection, I think, is going to be more and more made using AI, because, biological features can be complex features, right? Not individual mutations, but, maybe things like HRD, which, this BRCAness that I was telling you about, which is not not a single mutation, but in fact a collection of mutations that you kind of agglomerate into a particular feature across the entire genome. I think many more biomarkers, if you want, are going to be of that shape. They're going to essentially kind of combine multiple different sort of features or data points into essentially a prediction about response to a particular therapy. And I think that's where AI is really going to play a key role, is, to establish this relationship. And you can sort of generalize this even further, because if you really think about how we should treat patients, right, especially in the context of this complexity, it's not obvious to me that we should treat patients one drug at a time. We should really be thinking about drug combinations since at the beginning, right? Now we've had many sort of for many patients, we're thinking about, with try to treat them with one drug, and it works maybe for a certain amount of time, then it stops working, and then we move on to another drug, and then the same, it kind of stops working, and we move on and move on and on. That's also partially due to the fact that we understand what drugs do individually, right? But we often don't understand what they do together. I do think that's where AI also should play a role, is to kind of give us make us able to predict how to accurately combine existing drugs, that we can achieve synergy, or we can achieve more efficient killing of tumor cells. And in fact, we're doing quite a bit of work in our lab actually on this. We're building deep learning models that can help identify drug combinations that are specific for every patient. I haven't shown anything on this here, but actually it seems to work reasonably well. We're able to build deep learning models that can take the gene expression profile of every patient. We've pre-learned a model based on expression profiles of thousands of patients. We captured essentially fundamental patterns of biology across many patients, able to generalize across patients in terms of patterns of biology that keep coming up over and over. Not individual mutations, but really like multiple genes being expressed at the same time. And we can relate that to basically the efficacy of drug combinations. And that, as I said, turns out to work actually surprisingly well. It's just another example of how we can use AI as a way to predict combinations that could potentially be effective also in patients. And we have to test the paradigm in patients, but in the preclinical setting, I think that seems to work really, really well. And I think it kind of points to the idea that, again, in the future, we're going to measure things, we're going to have these machine learning models, and we're going to make predictions about not just individual drugs, but maybe combinations of drugs, maybe even, how to sequence the combinations to essentially achieve optimal effect, optimal efficacy. I'm talking here about a very different form of medicine, which, as I said, is much more personalized and much more detail when it comes to assessing the biology of every patient, but also uses AI as a way to combine those different data points into specific predictions.
So I want to ask about, this is actually kind of a multi-part question, because it's about more like quality of data. Because from my rudimentary knowledge of AI, I know that you need to have good high quality data and a lot of data to train AI. So that's, poses a particular challenge in rare disease, to have that volume of data, first of all. That's kind of more unique to rare disease. Though I know you just mentioned we should really think about all cancers as each being rare disease. But also quality of data in terms of biases, like a kind of how to make sure we minimize and eliminate bias in data, as well as I think, something I'm just learning is a kind of a big issue, which is the difficulty of actually harnessing all the clinical data, because it's actually pretty freeform. But like it's in notes and it's not at all uniform. And that's really critical, like you said, for all the outcome information, or even just, treatment. So sorry, that's a long question with a lot of like a
no, that's great. Yeah, I love it. I would say there's multiple different components, to your questions, but different components themselves. So one is yes, every patient is a bit different, right? And so we think that there are commonalities when it comes to the underlying kind of biological pathways, right? We know that biology, is complex, but nonetheless, it kind of coalesces over specific pathways, which tend to be found and reused by cancers. Cancers are typically not fundamentally inventing anything new. They're kind of, co-opting existing pathways that are used during development, for example. And so, there's a limited number of them. It's just, it's a reasonably large number of them, but still, it's not unlimited. So we think that by analyzing multiple cancers at the same time, and not just cancers, but also cell lines, organoids, essentially having a lot of data points, we can capture a good set of biological kind of patterns. So let's say pathways that we kind of build the building blocks of cells, if you want, in some ways, or the higher level building blocks. I think your question was also related to how we learn, right, these AI models. So one is, diversity of patient profiles. And so when we build AI models, and especially coming from patients' data, we have to make sure that we don't propagate, biases that, may exist in patient populations that are treated by, in in the current medical system, which has its own biases. And so that's very much at the level of the construction of the AI models. We have to make sure that the data that goes into training these models, is free of biases, or at least that we know the biases so that we can correct them. And the last component of your question was focused on the clinical data, right, and the quality of it, right, and how we can use it, to learn how to connect biological patterns with outcomes. And that's the fundamental limitation. I think even more than just that limitation, I think it's just also the fact that it's limited. There's not a lot of it. And frankly, that's also a big motivation for us for this, kind of approach here to do tumor organoids, to build these avatars of patients' tumors, and then do drug screens to kind of test, let's say 150 different drugs on every single patient sample, right? Is that we think that gives us a lot more data points on the patients. I mean, we don't treat the patients, we treat the avatars of the patient if you want, but we get a lot of data points on these patients. And we think that these data points can in turn be used to train the models and to consolidate the models. So I think these AI models of the future that predict outcomes, they'll be trained based on patient data, with some of the limitations that you mentioned, but we'll make a lot of use of data coming from avatars as well, because, it's quite good. The models, the organoids themselves are very similar to what you see in the patients. And so you get a lot of data points coming from these models that you don't get from the patients. And so that kind of, allows us to compensate for, the limited clinical data that you get from patients, by essentially adding a lot more data that should go into training models. And that's kind of, a little bit, so what I started showing here, even though this is actually not using clinical data directly, but it's using this high throughput drug screening data as a way to train models to predict efficacy of individual drugs. I don't know if that answers your questions, but that's the best I could do.
Yeah, no, that was impressive to kind of address all of that so succinctly. So maybe one last question to leave you with, which is, so assuming, we get over that hump of the quality and volume of data, another big opportunity kind of afforded by AI is being able to integrate all the different data types, because as you mentioned in the beginning of your talk, there has been this kind of almost data explosion, with all these new technologies. And so it's, not just DNA, but it's the RNA and proteins and epigenomics and kind of really being able to integrate all of that. And so just practically speaking, and I know all your work is really applicable to across cancers, but selfishly, I like thinking about how the osteosarcoma community can kind of harness this. How does, if you're a researcher working in a lab and your focus, you're working on specifically, let's say, proteomics or something, how do we start to kind of plug everyone in to be able to contribute data to this kind of larger effort where we can start to integrate all the data into this model? Like, how do we kind of bring it all together? And again, as an individual researcher, how do I kind of plug into that effort?
Yeah, I mean, I think that one thing that we learned is that obviously there is something to be said about numbers, right? And so I think the rare disease community needs to do better is to team up, form consortia, to be able to collect data at scale across, a good number of patients, so that you can learn from, multiple different, let's say, profiles, of patients, and really kind of, try to generalize. It goes beyond this, right? Because I think there's this idea that, once you find subtypes of tumors, you're kind of done, right? But I think we know that the biology of patients is quite unique. But I would say one thing that's also reasonably clear is that you don't necessarily need to model the entire biological features of a patient. You can potentially kind of, model a subset of them. The point here is that I think there's a lot to be said in the rare disease space, obviously about patient-derived models, sort of taking, cells from one patient and growing them and being able to make a model for that one patient. But if you can't do that, maybe look for models that share biology with the patient that you're looking at, to be able to transfer information from these models with cell biology towards the patient that you're looking at. So the point about this is that there's strength in looking at large cohorts, even though again the patient's biology is going to be quite unique and all that. So now the reason I'm saying this in the context of your question is that I think if somebody is doing proteomics of osteosarcomas, right? It's more powerful to look at that data in the context of a large cohort. So ideally being able to have enough tissue across multiple institutions to be able to do proteomics in a larger cohort, will inform the scientists more on basically the biology in general of osteosarcoma. But I think one of the point of this presentation is also to say that just looking at proteomics in isolation, it's not going to give you the whole picture, right? So I think it's important to keep that in mind. I think the future is really towards this kind of multi-omic characterization. I think you really have to think hard about this. There's a lot of proteomics that's done from bulk tumors, right? And when you do proteomics as an example, when you typically assess proteins coming from every cell, and if as I showed in the context of the B-cell lymphoma that I showed briefly, the minority of the tumor cells are the minority of the cells, right? I mean, you're actually doing proteomics mostly on the non-tumor cells, and so the interpretation of that is obviously critical. But that's when having more information about the cancer and knowing that you have lots of non-cancer cells in there, is important in terms of interpretation of another modality. But the future is really towards multi-omic characterization. I think at multiple level. I think what I showed you is these 3 layers, right? You start with the DNA, which is just the tumor cells, and you go 1 layer up, and you start seeing the cells, including the tumor cells, but also what's around them. And then the layer above is basically being able to take the cells and kind of poke them and kind of see what else they reveal by themselves. I think that in its totality gives you a lot of information about every patient, and I think that's the kind of information that we need to be able to understand the disease within every patient. So whether it's proteomics combined with maybe a single cell analysis or maybe DNA sequencing, I think these are things that need to be thought about. I think there's a lot of potential, for example, with proteomics to find surface markers, maybe novel targets that could potentially be used to create antibodies with chemicals attached to the antibodies that can specifically kill the tumor cells by bringing chemotherapy into the tumor cells. These antibody drug conjugates. So it kind of depends on how you want to use the modalities, but context matters, and I think often individual modalities don't give you enough information.
Well, thank you so much. You've given us so much food for thought today, and I think
good.
a topic that can inform everyone's work. So thank you so much for joining us today, Dr. Elemento on OsteoBites, and for making it better for cancer patients. More information on this and all OsteoBites can be found on our MIB Agents YouTube channel, on our website at mibagents.org, and at your favorite podcast place. And next week on July 18th, we will drop a new episode of Osteo, which is our AYA podcast. And in our July episode, our host Mia is joined by Osteo siblings Dr. Chris Kuo from CHLA, Matt and Valerie to discuss the often forgotten and overlooked children in an AYA cancer diagnosis, who are the siblings. More info in the chat about that, as well as a link to a fantastic article that Dr. Kuo published recently about the sibling experience. And then we hope you can join us the following week on July 25th. We will be back with OsteoBites with Dr. Bhuvana Sethi. She's a pediatric hematologist oncologist at Nationwide Children's Hospital, and she will be joining us to discuss a clinical trial called a multi-institution study of TGF-beta imprinted ex vivo expanded universal donor NK cell infusions as adoptive immunotherapy in combination with gemcitabine and docetaxel in patients with relapsed or refractory pediatric bone and soft tissue sarcomas. I think that might be the longest clinical trial name I've come across, though it has been nicely shortened to TINKS, which is the acronym for that trial. You can find our OsteoBites lineup for the next few months on our website, And if you have any ideas for future topics you'd like to hear about, please feel free to let us know at events@mibagents.org. Thank you again, Dr. Elemento, for spending an hour with us today. And thank you also, Daniel, for joining us today. We hope to see you all back here on July 25th when we chat with Dr. Sethi about the TICS trial. Thank you. Thank you so much, Dr. Elemento. Thank you, Daniel.
Thank you. Thank you for having me.
Guys. Okay, have a great rest of the day.